Good afternoon. I'm Dr. Ron Victor. I'm a cardiologist at Cedars-Sinai. You can tell because I've got an EKG on my tie. And I'm a great friend of Dr. Rochelle Crosby Watson, who I want to congratulate for this fantastic course. And I have a joint appointment here at UCLA. This is the first of 3 mini lectures about the nitric oxide pathway as a putative new drug target for treating patients with Duchenne Becker muscular dystrophy. Yeah, I know you've seen a slide like this. You know about the clinical course of this terrible muscle wasting disease. And the reason I'm showing this is that although basic research in muscular dystrophy has flourished, clinical translation has not. So the disease gene was discovered as dystrophin over 30 years ago. That has led to no specific drug or no specific therapy of this terrible disease. Now, as an adult cardiologist, not a pediatrician, not a neurologist, I had been on the fringes of this field for a few decades now, until very recently, where I have really started to delve into clinical trials. And it seemed that until very recently there were almost no clinical research in this area. But that's all changed. The landscape, fortunately, has changed dramatically, and now there's a groundswell of clinical trials that are either enrolling now, recently completed, or about to start in the next few years. And the experimental strategies these trials are testing are as follows. First is to restore the missing protein dystrophin, or upregulate the dystrophin homolog utrophin. In addition, in terms of signaling abnormalities that are downstream, to increase contractile muscle mass, reduce inflammation and fibrosis, correct abnormal calcium handling, and finally to correct the abnormal blood flow regulation that I've been studying now for really 30 years. Wow. So the outline for this talk is as follows. I want to explain the rationale for the nitric oxide cyclic GMP pathway as a new drug target to treat dystrophinopathy. The concept that PD5 inhibition with drugs like with erectile dysfunction drugs like Cialis or Viagra, why on earth they would be used to being repurposed to treat Duchenne. We've done that in proof of concept studies in patients just in the past few years to show that PD5 inhibitors and nitric oxide donors can improve the blood flow abnormality in adult men with Becker and in pediatric patients with Duchenne. And those studies then have teed up clinical trials to see if better blood flow will actually preserve muscle. Okay, so this is the cartoon of the dystrophin glycoprotein complex. You know that dystrophin, this long, big protein, you've seen this cartoon, I'm sure now, many times, that this is a structural protein that provides a physical link between the actin-based cytoskeleton and the extracellular matrix. As a cardiologist, who's not a neurologist, I see this as a shock absorber that prevents tears in the membrane during activities of daily living and much less very heavy physical exercise. But in addition, dystrophin is also has a signaling function. It targets a number of associated proteins into combinatorial relationship right at the sarcolemma. And among these is my favorite protein, which is nNOS. And I'm very proud of Rochelle for getting the binding sites exactly right. So nNOS is highly enriched in skeletal muscle. This is a splice variant of the neural isoform, but it's not hooked up to neurons. It's hooked in muscle. It's hooked up directly to the muscle membrane by its association with dystrophin. It binds both to spectrin-like repeats, these spring-like repeats called SR1617, and also to the adaptive protein alpha-syntrophin. That does two things. Because this is a calcium-dependent enzyme, the calcium signal that causes the muscle to contract causes coordinate activation of nNOS, and the nitric oxide release is right at the sarcolemma, where it has a very short, this is a gas, right? And it has a very short diffusion distance to get to microvessels where it can dilate microvessels. It's a way that the muscle can regulate its own blood supply. So, to recap on this, this inset, nNOS mu is this neuronal nitric oxide synthase splice variant that's only enriched in skeletal muscle and in cardiac muscle. It's muscle specific, and this is by far the main source of nitric oxide in mammalian skeletal muscle. It's targeted to dystrophin specifically, or targeted to the sarcolemma specifically by this SR1617 binding site. You'll see why that becomes important in a bit later. Again, it's the main source of nitric oxide in skeletal muscle. It's one of the key ways that the muscle can regulate its own blood supply. The enzyme is not active when the muscle is resting, and it gets activated during exercise, so that the amount of nitric oxide produced is greater to the intensity of the muscle contraction. So this is the concept of functional sympatholysis, and I need you all to understand this to go forward. So I live in West Hollywood. Imagine I'm in a spin class, and I'm seated on a bicycle, and my legs are exercising, and my arms, if I'm doing it right, not even holding on. So my arms are resting, my legs are exercising. So with graded exercise, the sympathetic system turns on from the brain and goes down to the resting muscles to release norepinephrine and cause alpha-adrenergic vasoconstriction. That shunts the blood flow from the resting muscle to the metabolically active exercising muscle. And if there were alpha-adrenergic vasoconstriction in the exercising muscle, that'd be counterproductive from a teleological standpoint, because it would choke off the blood supply to the exercising muscle. Well, what happens is there are metabolites produced locally by the exercising muscle that negate alpha-adrenergic signaling. And to summarize about 20 years of research, the main metabolite from the exercising muscle is nitric oxide derived from sarcolemmal nNOS. May not be the only factor, but all of our work and from multiple labs suggests that it's the major factor. So this is a cyclic GMP-dependent process by which the downstream alpha-adrenergic signaling is attenuated. So that prevents the sympathetic nerves from choking off the blood supply of the working muscle and optimizes muscle perfusion. And that metabolic modulation of alpha-adrenergic vasoconstriction is a mouthful, and so that's why we call it functional sympatholysis. Okay, so that's a normal situation. And so Gail Thomas was a postdoctoral fellow in my lab at UT Southwestern in the early 90s. And so we wanted to study, this is functional sympatholysis in an anesthetized rat. And this is the first paper we published on this. And so what you see is the rat's anesthetized, and there's a Doppler flow probe around the femoral artery going to the active muscle. And so here's the panels are arterial pressure, femoral blood flow, and force at rest, and then doing intermittent tetanic electrically induced contractions of the gastrocnemius muscle. The gastroc in the calf is a fast twitch muscle, and it rapidly fatigues. So at rest, we stimulate the lumbar sympathetic chain going electrically stimulated to that gastrocnemius hind limb. And what you see then is that causes an increase in blood pressure, a decrease in femoral blood flow. So increased blood pressure, decreased flow is vasoconstriction. Well, then we electrically contract the muscle, and you can't see there's the intermittent contractions because of the sweep speed on this polygraph. But what you see is that we stimulate the muscle to contract, it starts to fatigue, blood flow increases, that's active hyperemia, and then when we superimpose about halfway through the same degree of sympathetic stimulation when the muscle is working, the blood flow doesn't go down, it actually goes up, and that's functional sympatholysis. This is not active vasodilation, it's loss of sympathetic vasoconstriction. It's a passive increase in flow to match the increase in blood pressure. And if you look, there's a slight restoration of force output during that, so the muscle likes it. So the idea is that improved blood flow, it has an anti-fatigue effect. So that's the original record. And I just want to say that Gail Thomas and I have been scientific partners for several decades now. I'm very proud. Gail is a full professor at Penn State in Hershey. So These are the summary data from Gail's initial paper. So this is the summary data from a large number of rats. So with sympathetic stimulation and resting muscle in blue, there's a big vasoconstrictor response. Conductance, which is 1 over resistance, goes down by about 60%. And all that vasoconstrictor response is wiped out when the muscle is contracting. This effect is not due to it could be due to the fact that maybe the sympathetic nerves can't release norepinephrine. It's not that kind of effect, because when we infused exogenous norepinephrine right into the blood supply of that muscle, we reproduce the same sympatholytic effect. And we've done multiple additional studies to show this is a downstream effect on alpha adrenergic receptors. So that was in the rat, and the question is, where was the nitric oxide coming from? So was it endothelial nitric oxide in the lining of the blood vessels, or really is it skeletal muscle sarcolemmal nNOS? So it took us about a year to miniaturize techniques from rats to mice, because they're so darn small. And so this is the same basic setup as in the rat. And in an MDX mouse, which is the standard mouse model of Duchenne, what you see is that, again, this is actually norepinephrine infused. It's not electrical stimulation. It's close arterial infusion of norepinephrine. So there's an increase in blood pressure, and blood flow goes down, and femoral blood flows down. It's vasoconstriction at rest. And during the fatiguing contraction, there's essentially no sympatholytic effect. The increase in blood pressure is accompanied not by an increase in flow, but by abnormally persistent decrease. The bottom tracing is cut off a bit, but what we showed was that we had exactly the same phenotype in the nNOS null mouse. So this was really exciting. And it was the first paper we published in the Proceedings of the National Academy of Sciences in 1998. So this is impaired sympath in the MDX mouse and in the nNOS null mouse. So then we were really off to the races. And they had the summary data from those experiments just showing that in black bars are the vasoconstrictor response in resting mouse muscle, MDX or nNOS, and then exercising in white, and you can see there's essentially no attenuation of the response during muscle contraction. So that was, let me stop here. This is back in 1998. We actually extended this work to boys with Duchenne. I'm going to breeze over those earlier studies because I want to fast forward to the new work, which is more translational. So we knew there's impaired sympatholysis. We knew it seemed to be due to failure of sarcolemmal nNOS, and I didn't know what to do about it. We thought, and so that was about, fast forward about 10 years, and I was in Dallas trying to decide if I was going to take the offer to move to Cedars-Sinai, and this is just right before the first of the year. This paper came online. Yvonne Kobayashi, who's shown in this picture here, was a junior faculty member in Kevin Campbell's lab at the University of Iowa. This is just a fantastic group in muscular dystrophy, and believe that Rochelle trained at the same time as Yvonne in this fantastic lab. So what Yvonne did was she said that she found that a single dose of Cialis or Viagra, common PDE5 inhibitors, before exercise would prevent post-exercise fatigue and prevent blood vessel spasm, and she interpreted the data based on our work. And I went, why on earth didn't we do that before she did? She beat us to it. We thought about it a little bit, and I didn't think it was going to work because I didn't think there was any. What I thought was that when nNOS is in the sarcolemma, is not in the sarcolemma, it goes in the cytosol and is chewed up by proteases. My understanding was there was essentially no nitric oxide produced, but that's not the case, as we'll see. And so this study changed my life. This came out right at it says November of 2008. I didn't see it until December, and we quickly sent a grant to the Muscular Dystrophy Association because, so, well, these are mouse doctors. They're great mouse doctors, but they're not going to do it in humans, and we have all the techniques to do that. So this is Yvonne's handiwork, and what it shows, this is a laser light beam just at the bottom of a mouse cage in the animal care facility at the University of Iowa. And so each dot is when the mouse stands up on its hind limbs. You know, mice are very twitchy. And if you look in the animal care facility at nighttime when they're awake, these nocturnal creatures are awake, you can see they stand up on their hind limbs, twitch their nose a bunch, sit down, do this, a whole mess. And so every time they stand up on their hind legs, it crosses the laser beam and counts 1. So pre-exercise, the wild-type mouse, the C57, is very twitchy before exercise. They put the mouse on a downhill treadmill. And you know, we'd never do that to patients because from day one, the parents are taught, don't let your child with Duchenne do downhill exercise because it stretches the muscle rather than contracts it. And that accentuates the tears in the membrane in Duchenne. Well, anyway, there was no load downhill exercise for 10 minutes, put the mice back in the cage, and the wild-type mice are still twitchy as can be. The MDX mouse is very active before exercise, but after the same bout of exercise, the mouse just sits in the cage as though they're fatigued. Well, you can't ask the mouse if she's fatigued, but that's what it was interpreted. So, and then here are the vessel spasm. This is a immediate post-mortem angiogram. And what this is, these are the microvessels inside the muscle that are feeding the muscle with blood and oxygen. And you see these sausage-like spasms. That's vasospasm in the feed arteries of the hind limb of the mouse, in the untreated mouse. And if one single dose of tadalafil, Cialis, 3 hours before the study, and almost all the vessel spasms are prevented. And I didn't show it, but it recreates the normal post-exercise activity phenotype and prevents the post-exercise fatigue, if you will, or inactivity to be more precise. So how do we translate that? Is it that there's, let me go back here, the spasms would be like might happen in a patient with peripheral artery disease that has claudication. In other words, they're fine at rest, but adult patients with a lot of atherosclerosis and have, especially diabetic patients, for example, will have, because the blood supply is just enough to cover the low metabolic demands of the muscle at rest, but during the activities of daily living, patients get real aches in their calves and have to stop exercising and let that go away and do it again. It's like angina in your legs. So we think this is microvascular angina, and could it be, or in the lower extremities? So could it be that that's why the mice weren't moving around? Well, to translate this, we first thought we'd turn to patients with Becker muscular dystrophy. This is a milder form of Duchenne. These are in-frame mutations, so the reading frame is maintained, whereas in Duchenne, those are out-of-frame mutations. So some dystrophin is made. It's often not a reduced amount, but the key thing is that it's an abbreviated protein. It's shortened dystrophin. And so many, but not all the mutations, delete the very exons that encode the SR1617 nNOS binding site. So the nNOS, in those patients, nNOS is displaced from the sarcolemma to the cytosol where it's not supposed to be, where I believe it's chewed up by proteases, and the amount's not reduced 100%, but about 95%. So you're left with about 2 to 5% of the normal amount of protein, and it's in the wrong place. So the question is there any nitric oxide leaking out of the muscle cells? So why don't we start with Becker? Well, first of all, these tend to be 30 to 40 or 20-year-old adults, and we thought from an IRB perspective it would be easier to get this approved before the pediatric patients. And because these are adult men, it's very easy to work with these individuals. So in addition, the fact that some of the patients they don't have the dystrophin backbone to target nNOS to the sarcolemma, and others do, it's an incredible opportunity to have, if you will, muscle-specific mutations in adult human beings who've had this problem for their entire life. So an incredible opportunity to learn about to test our hypotheses. Okay, so here's the idea that in the Becker skeletal muscle cell, that there's a short dystrophin that can't bind nNOS. So nNOS is misplaced to the cytosol, where the idea is that perhaps there's a little bit of residual NO that leaks out of the muscle cell and gets to the interstitium where there's vascular smooth, where the vascular smooth muscle feed arteries are. The NO then increases cyclic GMP, which, because if that's attenuated, then we have unopposed sympathetic vasoconstriction, maybe impaired dilation and functional ischemia. And then tadalafil the question is, can tadalafil boost that downstream signal by inhibiting PD5, which chews up cyclic GMP? So the point is that Cialis does not increase NO production, it boosts the downstream effects. Okay, so this is the first paper I published in my lab at Cedars-Sinai. I've been here 6 years. And this is Beth Martin, who was the first author on this paper. So this shows that tadalafil alleviates muscle ischemia and rescues functional sympatholysis in adult men with Becker muscular dystrophy. So Beth started with a case control study and then an acute treatment trial. So these are the participant characteristics. We studied 10 patients, mean age of 37. They had a normal body mass index, normal blood pressure, normal left ventricular ejection fraction, which means they didn't have severe cardiomyopathy. And MVC is maximal voluntary contraction, it's the maximal grip strength. So we asked the patients in the normal healthy controls to squeeze as hard as you can for a couple of seconds on a hand grip dynamometer. And they do that 3 times, and we take the highest level, and that's their maximal voluntary contraction. So the only difference between patients and the controls were, as you might expect, the patients were half as strong as the control subjects. Otherwise, they were well matched for age and gender, of course, and for all these other factors. Okay, so here's the experimental setup. This setup took us 5 years to develop after we did the initial rat studies. I had a couple of really bright cardiology fellows from the University of Copenhagen in Denmark, and especially Jim Hansen, and he worked like crazy for 5 years to make this work, just to show you how hard it is to translate work from rats to patients. So the way this works is we tried to mimic Gale's setup in the anesthetized rat, and instead of having electrical stimulation, well, first of all, instead of electrical stimulation of the sympathetic chain, which of course is not, ethically feasible at all in a human patient, we use this lower body negative pressure thing, and this stimulates orthostatic stress and causes reflex sympathetic activation. This is basically a garbage can hooked up to a vacuum cleaner, well, it sort of gave me tenure at UT Southwestern, so I call it my tenure box. Anyhow, we reduce central venous pressure with this thing to simulate orthostatic stress, but the patient or subject is still lying down, so it doesn't drive all our instruments crazy. And what's cool about it is that it basically triples sympathetic outflow to all the skeletal muscles in the body. It's a very reproducible stimulus. It's very important for studying attenuation of that reproducible stimulus. We can record the sympathetic traffic going to the skeletal muscle with microelectrodes. And then instead of using Doppler flow probes at first to measure blood flow, we use near-infrared spectroscopy. They're these non-invasive probes that are placed on the skin over the flexor digitorum profundus. That's the muscle, that's main muscle of hand grip when you go pump gas at the gas station. And the way this works is that laser light in the near-infrared spectrum in the 700, 900 nanometer range is absorbed by the heme porphyrin complex of hemoglobin and myoglobin. And there's a slightly different chemical shift in the oxygenated and deoxygenated species. So this allows us to measure oxygen supply versus demand right at the microcirculation level, which is where we think sympatholysis works, not in large arteries, right in the microcirculation, the intramuscular microcirculation bed. So in a resting arm, if we turn the suction box, you see in the cartoon that there's a decrease in hemoglobin, myoglobin deoxygenation. And we turn the box off and it comes back to baseline. The question is, if we exercise the arm, will we prevent that vasoconstrictor response? And so this is the hand grip dynamometer. The subjects do rhythmic hand grip. It's 1 second on, 1 second off. They get their maximal grip strength. Then on the computer screen, we set a target line at only 20% of their maximum, 1/5 of their max. This is very easy exercise. Even a weak patient with Becker or Duchenne can do this. So it's squeeze, relax, and that's what we're doing. Okay. So this shows that sympatholysis is impaired in a patient with Becker muscular dystrophy. So here's a healthy control in the top panel A. And what you're seeing is this is the muscle oxygenation, tissue oxygenation signal by near-infrared spectroscopy. The bottom is the force output. So if we turn the lower binegative pressure box, our sympathetic stimulus, and you can see there is a decrease in muscle tissue oxygenation, then we turn the box off with the arm resting. Then we have the patient, this is the normal subject, squeezing at 20, off and on at 20% of his maximum. And there's the muscle tissue oxygenation decreases and reaches a new steady state because the muscle's consuming oxygen. Then we superimpose the lower binegative pressure, LB and P, and there's no additional deoxygenation. That is functional sympatholysis in a human forearm. We then have a blood pressure cuff around the upper exercising arm, inflate it to suprasystolic pressure, like 200 millimeters of mercury, and that's to cause complete ischemia, shut down all blood flow to that forearm, and we then see the nadir in the oxygen signal. We then let the person stop exercising, take the cuff down, let their arm come back to normal, and there's a big, the oxygen signal comes roaring back to baseline, overshoots the baseline. And so the difference between the nadir in the oxygen signal and baseline we call the total labile signal, and we use that to express all the functional responses as some percentage of this maximum deoxygenation. In the patient with Becker below, what you see is that at rest, the vasoconstrictor response, if I call it that from now on, looks just like it does in the healthy control. The force output is less because the patient is half as strong as the control subject. When he exercises, the oxygenation signal decreases. When we superimpose the same level of our reflex sympathetic stimulus, there's no attenuation of that vasoconstrictor response during exercise. That is impaired functional sympatholysis. We call that functional muscle ischemia. And if you look at the bar graphs on the right, in 7 healthy controls and 10 men with Becker, you can see that there's about a 60% attenuation in the vasoconstrictor response from rest to exercise in healthy controls. There's no attenuation in the mean data in the patients. So that was very exciting. And then we did a randomized double-blind crossover trial of tadalafil versus placebo, and this was really exciting. So the tadalafil is bought from Eli Lilly. It's a pill, and we had our research pharmacist hide it in an opaque capsule, so neither we nor the patients knew if they were getting active drug or placebo. And so they come in and get their baseline study. They get one drug in random order. They go home for at least 2 weeks to wash out the drug. The half-life of tadalafil is 17 hours. So a few days later, it's long gone, and 2 weeks later is more than enough time. Then they came back and got the other drug in case, again, some got placebo first, some got tadalafil. So here's a different patient than I showed you on the previous slide. With placebo, there's vasoconstriction at rest, preserved vasoconstriction with exercise. This is impaired sympatholysis, functional ischemia, and tadalafil fixes this patient right up in terms of blood flow. So the vasoconstrictor response is now attenuated with exercise. And we don't have to worry about the different levels of exercise because they're the same in this patient. It's a paired comparison. And if you look at the summary data, then the tadalafil rescued sympatholysis so that the exercise-induced attenuation of this vasoconstrictor response by near-infrared spectroscopy looks indistinguishable from the normal situation. So that was great. Now, it's possible to hide a lot of warts in mean data. So this slide shows we plot the individual data for each of the 10 patients on placebo and on tadalafil. Well, tadalafil's up top, placebo down below. And so these are the patient numbers, and the exons deleted are shown above the bar graph. So the black bars are the vasoconstrictor response at rest, and the white bars are the vasoconstrictor response during mild rhythmic hand grip exercise. So if you, in general, so first of all, the 45 through 48 deletion is the most common mutation that we see in ambulatory patients with Becker muscular dystrophy. And this exactly are the exons that seem to encode the SR1617 nNOS binding site. So we were pretty sure that none of these patients could bind nNOS to the sarcolemma. And some of others have different mutations. It'd be hard to predict a priori whether or not they could bind nNOS. But if you look on tadalafil, almost all of the patients, with a few exceptions I'll explain, the white bars are about half the size of the black bars, and that means functional sympatholysis. Without, on placebo, the black and the white bars are virtually all the same size, with one exception, and so that's impaired sympatholysis. So what I love about this is that the individual data show that the mean data are really representative. Now, there are two exceptions, and one of the things very important in patient research, especially when we're dealing with rare diseases, is to treasure our exceptions. So patient 9 has a huge number of exons deleted, all the way from exon 14 through 44. If you believe that, and Dongshen Dewan's data seems to suggest this from the University of Missouri, that the nNOS binding site is 45 through 48, then this is south of the nNOS binding site, and you should still have preserved sympatholysis, still nNOS binding. Well, this patient had preserved sympatholysis both on tadalafil and on placebo below. And then patient 10 has a deletion of 3 through 7. This is a very severe deletion. It's associated with a severe phenotype. This boy was initially diagnosed as having Duchenne because he had such a severe disease. When he was still walking at age 15, he was reclassified as Becker. But he's in this intermediate group. Either he has very mild Duchenne or very severe Becker. But he had clearly impaired sympatholysis and was the only non-responder in these first 10 patients. So we were worried, well, maybe what we're seeing about correcting the blood flow abnormality in Becker won't work for the more severe Duchenne. We'll come back to that. But I want to show you these. So in terms of the preserved sympatholysis in the patient, we sent this paper to Science Translational Medicine, where we had favorable reviews, but one of the reviewers very fairly said, all right, if you want us to publish this paper in this good journal, you're going to have to bring these patients back and get some biopsy tissue and show us that the patient with the preserved sympatholysis has preserved nNOS binding at the sarcolemma. Okay, so this is Rita Barisi, and this shows the histology, the histopathology in dystrophin staining in healthy control and in two of our Becker study patients. Patient 5 has the common 45 through 48 exon deletion. Patient 9 is the young man with the 14 through 44 deletion that had preserved, he was the one patient that had preserved sympatholysis without tadalafil. So Rita was introduced to me by Rochelle. They were both in the Kevin Campbell lab, and Rita is the world's expert on nNOS staining of human skeletal muscle. She directs the rare disease muscle biopsy lab at Newcastle on Tyne, and so she analyzes all the rare disease muscle biopsy specimens for the entire United Kingdom and has the best nNOS assay on the planet. apparently it's much more difficult to get good nNOS staining on human muscle tissue than on rat and mouse muscle tissue. So what we're seeing here, the H&E, this is normal subject, has pretty uniform sized muscle cells. You count, if you have to look closely, you can see that there are nuclei in a sarcolemmal location. And there's dystrophin C-terminus staining, which you expect to see. In the patients, the two patients with Becker, the histology is classic. There's a lot of replacement of muscle cells with fibrosis and fat tissue. There's central nucleation. The cells are swollen, and they're in different sizes. They both have some dystrophin because they're staining for dystrophin, the C-terminus of dystrophin. This proves that we're studying patients with Becker and not Duchenne. Okay. So here's the key experiment. So this is the nNOS staining at low power and high power on the microscope. So the healthy control has plenty of nNOS staining around the sarcolemma. The key is that the patient 5 who has impaired sympatholysis indeed can't bind nNOS to the sarcolemma. There's not any nNOS around those muscle cells in a sarcolemmal pattern. And that's expected because the 45 through 48 deletion bashes, if you will, the SR1617 nNOS binding site on dystrophin. Patient 9 that has the 14 through 44 deletion and has preserved sympatholysis has a preserved thin rim of nNOS around the sarcolemma. And when I got the email from Rita, it was labeled ta-da, because that makes the point. So there's sarcolemmal nNOS on patient 9 and not on patient 5. So it's pretty cool that in these two patients, at least, sympatholysis follows sarcolemmal nNOS. Now, in addition, you see this brown staining that's labeled cytosolic nNOS. There are a bunch of papers published by other investigators suggesting this brown, this staining with the antibody means that there's nNOS in the cytosol of these patients, and presumably that's the source of the muscle-produced NO that we can boost the downstream cyclic GMP signal with tadalafil. Rita isn't 100% sure that this isn't just a staining artifact, but she's cautiously optimistic that it really is cytosolic nNOS, but important to be a bit skeptical. So we don't know for sure, and so went back to Gail's mice and tested the concept. And what we showed was that, what Gail showed in these studies, that tadalafil restores sympatholysis in MDX mice, but not in nNOS null mice, because they don't make any nitric oxide. So here's sympatholysis in the wild-type mice and in untreated MDX mice. Again, the black bars are the vasoconstrictor response in the anesthetized mouse with the muscle resting, and the attenuated vasoconstrictor response showing sympatholysis when superimposed on muscle contraction. The MDX mice has no sympatholysis, and tadalafil fixes the MDX mice right up. The sympatholysis is clearly restored with tadalafil, whereas tadalafil has no effect on the nNOS null mouse. And so that's really nice, I think, key proof of concept for that tadalafil can boost a residual NO cyclic GMP signal generated by nNOS misplaced to the cytosol. Okay, so this slide shows that the tadalafil restores sympatholysis in boys with DMD, just like it did in patients with Becker. There are two doses. What there are here, the black bars are the vasoconstrictor response to lower body negative pressure in resting forearm muscle, and the white bars in exercising forearm muscle. On the right are the healthy controls, and in the middle are a dose escalation response to tadalafil. These are open label studies. They're not double blind, as we showed you in the Becker patients. And what I forgot to mention about the Becker patients is the dose we used was 20 milligrams, single tablet. That is the maximum allowed dose for erectile dysfunction in an adult male. So 0.5 milligram per kilogram in a child is roughly equivalent to the 20 milligram dose. The higher dose is equivalent to 40 milligrams, 1 milligram per kilogram, or 40 milligrams in an adult. This is a pulmonary hypertension dose, and while everyone sees on TV all the advertisements about Cialis and Viagra for erectile dysfunction, these drugs have been a game changer for patients with a very severe disease of pulmonary artery hypertension. This disease can come on in anywhere from neonates to young adults, and it causes progressive fibrosis within the lungs. And it takes twice the dose, about 40 milligrams at least, to have an beneficial effect. This improves exercise performance, exercise tolerance, and quality of life big time in these patients. It was really the first major breakthrough in pulmonary artery hypertension. So that there's precedent for repurposing PD-5 inhibitors for other diseases. And so here we have impaired sympatholysis in the boys with Duchenne untreated. And then tadalafil produces a dose-dependent correction in these boys. So if you look, the low dose causes about a 50% attenuation in reflex vasoconstriction during mild exercise that approximates that in the healthy controls on the right. The high dose produces a supernormal correction. So it's very exciting because it showed that our concern about whether this approach would work for the more severe disease of Duchenne as well as the milder disease of Becker turned out that in fact it did work. So that got us very excited about this. And this is a dose-finding trial that teed up a clinical trial. And then we wanted to look at post-exercise vasospasm. So Xu Tang is both a fantastic echocardiography technologist, but also a really bright research scientist in our group. And so remember Yvonne Kobayashi's study showing post-exercise vasospasm in the microcirculation of the skeletal muscle, the MDX mouse. To try to translate that, we use Doppler blood flow velocimetry over the brachial artery. Is that where we want to measure the blood flow? No, we'd like to measure it directly in the microvessels, not in these big conduit arteries. But it was an easy thing to do to start with. We are now going to use contrast-enhanced ultrasound to measure microvessel perfusion, but that's in the works. In the meantime, so this is blood flow by Doppler in the boys with DMD, and this shows the sample volume. That black horizontal line is the artery and the stuff within the muscle, and then what you see is the Doppler blood flow coming through below, and that's the systolic and a little bit of diastolic flow, right? But most of it's systolic, and that's in a resting forearm. This is post-exercise in a normal subject, and here you see that you see the flow is much fuzzier, and the area under the curve is much bigger. So that's post-exercise hyperemia. So if you look on the right, in the healthy controls, 10 healthy boys age 9, blood flow increases almost by 100%, so essentially doubles from rest to exercise. This is only 20% hand grip, but even there it ought to increase probably about 5 or 10-fold. And the reason it's not is that we're too upstream from where we'd really like to be measuring. In the untreated boys with Duchenne, they increase their blood flow by less than half of the normal amount from rest to immediately post-exercise. And then tadalafil produces a dose-dependent correction of that, but it takes the high dose to do it. Remember that it looked like the 0.5 milligram per kilogram dose was able to rescue some patholysis, and the high dose produces supernormal correction. Well, here it looks like the dose response is a bit different from post-exercise hyperemia. And I don't know if that's real or Memorex or because we have a blunt tool with Doppler ultrasound to be measuring this. Anyway, it went the right direction. And so we couldn't sort out whether we should use the lower or the higher dose, and the clinical trial uses 2 doses, that are a little bit less in each one of these. But basically, the idea is that with daily use, that we're going to use both of those doses in the clinical trial. All right, so that's the end of this section. And if there are no questions, we'll move on to the next one. Thank you very much.